Chapter 10 Cell Reproduction 10.1 Cell Division. By the end of this section, you will be able to do the following. Describe the structure of prokaryotic and eukaryotic genomes. Distinguish between chromosomes, genes, and traits. Describe the mechanisms of chromosome compaction. The continuity of life from one cell to another has its foundation in the reproduction of cells by way of the cell cycle. The cell cycle is an orderly sequence of events that describes the stages of a cell's life from the division of a single parent cell to the production of two new genetically identical daughter cells. Genomic DNA. Before discussing the steps a cell must undertake to replicate and divide its DNA, a deeper understanding of Figure 10.1. A sea urchin begins life as a single diploid cell, zygote, that divides through cell division to form two genetically identical daughter cells, visible here through scanning electron microscopy, SEM. After 4 rounds of cell division, there are 16 cells, as seen in this SEM image. After many rounds of cell division, the individual develops into a complex, multicellular organism, as seen in this mature sea urchin. Credit A. Modification of work by Evelyn Spiegel, Louisa Howard. Credit B. Modification of work by Evelyn Spiegel, Louisa Howard. Credit C. Modification of work by Marco Bustraghi. Scale bar data from Matt Russell. Introduction. A human, like every sexually reproducing organism, begins life as a fertilized egg, embryo, or zygote. In our species, billions of cell divisions subsequently must occur in a controlled manner in order to produce a complex, multicellular human comprising trillions of cells. Thus, the original single-celled zygote is literally the ancestor of all cells in the body. However, once a human is fully grown, cell reproduction is still necessary to repair and regenerate tissues, and sometimes to increase our size. In fact, all multicellular organisms use cell division for growth and the maintenance and repair of cells and tissues. Cell division is closely regulated, and the occasional failure of this regulation can have life-threatening consequences. Single-celled organisms may also use cell division as their method of reproduction. Chapter Outline 10.1 Cell Division 10.2 The Cell Cycle 10.3 Control of the Cell Cycle 10.4 Cancer and the Cell Cycle 10.5 Prokaryotic Cell Division. The structure and function of a cell's genetic information is necessary. A cell's DNA, packaged as a double-stranded DNA molecule, is called its genome. In prokaryotes, the genome is composed of a single, double-stranded DNA molecule in the form of a loop or circle, Figure 10.2. The region in the cell containing this genetic material is called a nucleoid. Some prokaryotes also have smaller loops of DNA called plasmids that are not essential for normal growth. Bacteria can exchange these plasmids with other bacteria, sometimes receiving beneficial new genes that the recipient can add to their chromosomal DNA. Antibiotic resistance is one trait that often spreads through a bacterial colony through plasmid exchange from resistant donors to recipient cells. Figure 10.2 Prokaryotes, including both bacteria and archaea, have a single, circular chromosome located in a central region called the nucleoid. In eukaryotes, the genome consists of several double-stranded linear DNA molecules, Figure 10.3. Each species of eukaryotes has a characteristic number of chromosomes in the nuclei of its cells. Human body, somatic, cells have 46 chromosomes, while human gametes, sperm or eggs, have 23 chromosomes each. A typical body cell contains 2 matched or homologous sets of chromosomes, one set from each biological parent, a configuration known as diploid. Note: the letter N is used to represent a single set of chromosomes. Therefore, a diploid organism is designated 2N. Human cells that contain one set of chromosomes are called gametes, or sex cells. These are eggs and sperm, and are designated 1N, or haploid. Upon fertilization, each gamete contributes one set of chromosomes, creating a diploid cell containing matched pairs of chromosomes called homologous, same knowledge, chromosomes. Homologous chromosomes are the same length and have specific nucleotide segments called genes in exactly the same location, or locus. Genes, the functional units of chromosomes, determine specific characteristics by coding for specific proteins. Traits are the variations of those characteristics. For example, hair color is a characteristic with traits that are blonde, brown, or black, and many colors in between. 254 Chapter 10, cell reproduction access for free at openstax.org. Figure 10.3 There are 23 pairs of homologous chromosomes in a female human somatic cell. The condensed chromosomes are viewed within the nucleus, removed from a cell during mitosis, also called karyokinesis or nuclear division, and spread out on a slide, right, and artificially arranged according to length, left. An arrangement like this is called a karyotype. In this image, the chromosomes were exposed to fluorescent stains for differentiation of the different chromosomes. A method of staining called chromosome painting employs fluorescent dyes that highlight chromosomes in different colors. Credit: National Human Genome Project, NIH. Each copy of a homologous pair of chromosomes originates from a different parent. Therefore, the different genes, alleles themselves are not identical, although they code for the same traits such as hair color. The variation of individuals within a species is due to the specific combination of the genes inherited from both parents. Even a slightly altered sequence of nucleotides within a gene can result in an alternative trait. For example, there are 3 possible gene sequences on the human chromosome that code for blood type: sequence A, sequence B, and sequence O. Because all diploid human cells have 2 copies of the chromosome that determines blood type, the trait, is determined by the 2 alleles of the marker gene that are inherited. It is possible to have 2 copies of the same gene sequence on both homologous chromosomes, with one on each, for example, AA, BB, or OO, or 2 different sequences, such as AB, AO, or BO. Apparently minor variations of traits, such as blood type, eye color, and handedness, contribute to the natural variation found within a species, but even though they seem minor, these traits may be connected with the expression of other traits as of yet unknown. However, if the entire DNA sequence from any pair of human homologous chromosomes is compared, the difference is much less than 1%. The sex chromosomes, X and Y, are the single exception to the rule of homologous chromosome uniformity. Other than a small amount of homology that is necessary to accurately produce gametes, the genes found on the X and Y chromosomes are different. Eukaryotic chromosomal structure and compaction: if the DNA from all 46 chromosomes in a human cell nucleus were laid out end to end, it would measure approximately 2 meters, however, its diameter would be only 2 nanometers. Considering that the size of a typical human cell is about 10 micrometers, 100,000 cells lined up to equal 1 meter, DNA must be tightly packaged to fit in the cell's nucleus. At the same time, it must also be readily accessible for the genes to be expressed. For this reason, the long strands of DNA are condensed into compact chromosomes during certain stages of the cell cycle. There are a number of ways that chromosomes are compacted. In the first level of compaction, short stretches of the DNA double helix wrap around a core of 8 histone proteins at regular intervals along the entire length of the chromosome, figure 10.4. The DNA histone complex is called chromatin. The bead-like, histone DNA complex is called a nucleosome, and DNA connecting the nucleosomes is called linker DNA. A DNA molecule in this form is about 7 times shorter than the double helix without the histones, and the beads are about 10 nanometers in diameter, in contrast with the 2 nm diameter of a DNA double helix. The second level of compaction occurs as the nucleosomes and the linker DNA between them coil into a 30 nm chromatin fiber. This coiling further condenses the chromosome so that it is now about 50 times shorter than the extended form. In the third level of compaction, a variety of fibrous proteins is used to pack the chromatin. These fibrous proteins also ensure that each chromosome in a non-dividing cell occupies a particular area of the nucleus that does not overlap with that of any 10.1, cell division 255 other chromosome. See the top image in figure 10.3. Figure 10.4 double-stranded DNA wraps around histone proteins to form nucleosomes that create the appearance of beads on a string. The nucleosomes are coiled into a 30 nm chromatin fiber. When a cell undergoes mitosis, the chromosomes condense even further. DNA replicates in the S phase of interphase, which technically is not a part of mitosis, but must always precede it. After replication, the chromosomes are composed of 2 linked sister chromatids. When fully compact, the pairs of identically packed chromosomes are bound to each other by cohesin proteins. The connection between the sister chromatids is closest in a region called the centromere. The conjoined sister chromatids, with a diameter of about 1 micrometer, are visible under a light microscope. The centromeric region is highly condensed and thus will appear as a constricted area. Link to learning this animation illustrates the different levels of chromosome packing. Click to view content, https://www.openstax.org/l/packaged_DNA.